unicellular eukaryotes include the tiny, single-celled, either free-living or parasitic unicellular representatives of the domain eukarya. They would include protozoa and archaeozoa. Now, study of these single-celled unicellular eukaryotes often falls into the realm of parasitology. Parasitology is the scientific study of parasites, which include both the single-celled and multicellular parasites. Now, there are scientists that study unicellular eukaryotes that are microorganisms that are not parasitic, and oftentimes those individuals work in oceanography, zoology, and other areas of field biology. Two general groups of unicellular eukaryotes that we might review and discuss are the protozoa, which would be eukaryotic single-celled organisms with mitochondria, and archaeozoa, which are unicellular eukaryotes that don't have any mitochondria in their cytoplasm. Archaeozoa that we can find in nature may have intracellular bacteria functioning in the same fashion as the mitochondria would in a protozoan, and it is in fact a bit of evidence to support the theory of endosymbiosis. Archaeozoa may have no mitochondria or intracellular bacteria at all. These are anaerobic. They do not undergo cellular respiration with the use of oxygen, and as such, because they have a very different kind of metabolism, they really have no need for either mitochondria or intracellular bacteria. As we go through the various examples of both unicellular parasites and other types of microorganisms, please keep in mind what the mode of transmission might be. Frequently, many of our examples are going to be intestinal parasites that are transferred from what we call an oral fecal route of transmission. We get some form of infective stage of the microorganism or parasite on our hands or in our food or water, and then we ingest those infective stages, and then the parasite spends part of it or most of its life cycle in our intestinal tract. Even if it doesn't spend most of its life cycle in our intestinal tract, many of the parasites we will discuss have some portion of their life cycle passing through the digestive tract of mammals, which would include humans. Depending on where the parasite is located, always keep in mind what type of specimen should we obtain if we're looking for the various types of parasites we'll describe in this various sections of eukaryotic microorganisms, and what kind of diagnostic stage are we looking for in that specimen. Please note, all of the examples that I've chosen are clinically important and may have some photos or images that are very graphic in nature, and if they disturb you in any way or you find them unsettling, please disregard the images and just concentrate on the discussion and the text. The phylum Percolozoa, which is the modern classification group in which we would find the unicellular eukaryotes that have amoeboid cell shape and that move using pseudopodia, would also include as sort of a subtype or a generic description, Sarcodina. Sarcodina had been used many years ago as a classification grouping, but now Percolozoa is the more sort of correct terminology to be used. We can use either one. Percolozoa is the formal name of the phylum in which we would find the two species examples that we have illustrated on this page, Naegleria fowleri and Entamoeba histolytica. Now, both Naegleria and Entamoeba have amoeboid cell shape. and moved by pseudopodia, which simply means it has in a sort of active stage for motility very soft cell membrane structure as its outer covering, and pseudopodia are long extensions of the cytoplasmic membrane that allow this unicellular eukaryote to pull itself along in its environment. You can see examples of the pseudopodia sticking out, sort of emerging or extending from the cell body in the photo of Naegleria fowleri on the right. Entamoeba histolytica is a second example of a parasitic unicellular eukaryote. Entamoeba is the primary cause of amoebic gastroenteritis. So if we ingest the infective stage of entamoeba, we wind up with a very serious gastroenteritis. The typical symptoms, essentially dysentery or diarrhea due to entamoeba, isn't too unlike any other case of dysentery or diarrhea, and the distinctive differences between those two clinical conditions. Diarrhea we often associate with a loose stool, whereas dysentery can have either high volume with lots of fluids excreted from the intestine, or low volume with a lot of blood. In any case, the intestinal tract is highly inflamed, and it's a very sort of painful condition when patients have amoebic dysentery. Naegleria fowleri is the most common cause of what we call primary amoebic meningitis. Now, meningitis is simply a clinical condition with an inflammation of the meninges surrounding the brain. There are a wide variety of pathogenic microorganisms, including bacteria, viruses, and protozoa, and yeast, that can cause meningitis. Naegleria happens to be the most common cause of amoebic meningitis. There are a few other amoeboid species that can also cause human disease, but we're focusing on Naegleria for meningitis and then Entamoeba histolytica for gastrointestinal problems. Now, in this illustration, it shows us one of the common places that we might find Naegleria fowleri: in stagnant, essentially non-flowing or still sources of water. And there have been occasions, as example in this Roman bath in London, England, where tourists have decided to take a dip in the water in this ancient Roman bath. It really doesn't get a lot of freshwater exchange. I'm not certain if they're doing that now, but years ago there were a few tourists that decided to take a dip in this ancient Roman bath near London and did contract Naegleria fowleri, causing meningitis in those individuals. Now, unfortunately, with this kind of meningitis, it's often quite fatal. It's very difficult to identify quickly and difficult to treat. Primary amoebic meningitis simply means that the parasite has gained entry to the upper respiratory tract. Scientists are reasonably certain that once in the upper respiratory tract, the parasite migrates through essentially those very thin, bony layers at the back of the nasopharynx, through the tissues, through the sutures found at the cribriform plate in the skull, and then into the brain. Once it migrates into the central nervous system from the oral cavity or nasopharynx, then unfortunately this amoeboid Naegleria fowleri begins to grow and destroy central nervous tissue at a fairly rapid rate. Looking at the photos, you can see that the single-celled amoeboid parasites are not very distinctive. They don't have any sort of unusual shape. They're often described as fairly simple, amoeboid, eukaryotic cells, but they grow fairly quickly and, of course, do a tremendous amount of damage to the central nervous system. Even with intervention, mortality rate is extremely high, almost 100%, depending upon how quickly the pathogen causes destruction or infection in the meninges. The next group are the ciliophora. The ciliophora are unicellular eukaryotes that move using cilia. Cilia are small, tiny projections from the cytoplasmic membrane that contain microtubules. The microtubules are very much like garden hoses. They're hollow, they're very long, and they are associated in pairs on the internal structure of both eukaryotic cilia and eukaryotic flagella. The pairs of microtubules, we have 9 pairs around the periphery, just underneath the surface of a membrane structure, and a pair of microtubules in the center. We call it a 9+2 microtubule construction. In addition, there are tiny little molecular motors between each pair of microtubules, and the little molecular motors are called dynein arms, and that's D as in dog, Y-N-E-I-N. Dynein arms sort of look like little ratchets, and as they flex up and down, it causes the microtubules to flex, allowing these ciliophores to move quite rapidly through an aquatic medium. We have 2 examples of ciliophora, Paramecium aurelia, which is actually a non-pathogenic protozoa, and Balantidium coli, which is a pathogenic ciliate that causes essentially gastrointestinal upset and diarrhea. Paramecium aurelia is a free-living archaeozoa. By free-living, we mean it's not parasitic. It doesn't require to be inside or associated closely with another organism for growth. If we notice in this illustration, there's a magnified image of the power stroke of the eukaryotic cilia, almost like little swimming arms or oars on a boat. And in this single-celled organism, essentially performs all of the functions, or very similar kinds of functions, that your whole body does. It has a way of excreting excess water. It has that little pocket or groove on the undersurface in that illustration on this slide that is essentially a gullet where food is introduced. And if you recall, for archaeozoa, this organism doesn't have any mitochondria. It uses instead intracellular bacteria that perform a similar kind of function. Balantidium coli is a mammalian parasite that affects the intestinal tract. So we have essentially dysentery or diarrhea that can be caused by this ciliate. Notice in the photo on the right and the image on the left, we can see this is a fairly distinctive cell in terms of its nuclei. The nuclei of a Balantidium, if you get just the right perspective, often has an hourglass image or almost football-shaped image. And it's a fairly large ciliated cell. So when you have a stool sample from a patient or even a biopsy of the intestinal lining, they're fairly easy to identify compared to some of the other intestinal parasites.